DNA. So now let us talk about the enzyme. The most important enzyme is the restriction endonuclease, and we call it restriction modification system because this is a system which helps to modify as well as to restrict the entry of a foreign DNA, so that that particular DNA will either successfully replicate within the host cell or will be eliminated by the restriction system. So restriction and modification. Restriction is basically the endonuclease part by which the bacterium is capable of excising out or chewing up the foreign DNA that enters, mostly in the form of the phage DNA that enters into a bacterium. And if that bacterium is not the exact host of that phage, then that bacterium will restrict the foreign DNA by chewing it up by the endonuclease activity. But sometimes it can modify by methylating. The methylase enzyme is the modifying enzyme by which the bacterial cell can modify the incoming DNA by attaching these methyl groups covalently to the restriction sites where the restriction endonuclease has to act. Those sites are being modified by the system. So this entire system is called restriction modification system. There are 3 or 4 types of restriction modification systems in bacteria. So here this picture shows how this restriction modification system works. Here there are 2 strains of E. coli K and E. coli C, and there are 2 strains of phages, lambda C and lambda K. The lambda C phage is the actual lambda C, E. coli C is the actual host of lambda C phage. It is not the actual host of lambda K. So when lambda C infects E. coli C, it lyses all the cell with the efficiency of plating being 1, that is 100% of the cells are being lysed or get infected and lysed. But when these lambda C phages infect the other host, that is the E. coli K strain, they cannot successfully lyse the cell. However, at the rate of 10 to the power minus 4, that means out of 10,000 cells, one of the cells gets infected successfully and is lysed. That means in that cell, this lambda C DNA gets modified by the methylase activity of E. coli K strain, and the phages produced from those cells which are being lysed, if reinfect the E. coli K, they can lyse or infect the cell 100%. That is, the efficiency of plating is 1 in that case. That means those phages have been now modified and will not be restricted anymore in strain K also. So that means the restriction and modification system go hand in hand. So E. coli K strain, first time it restricts the most of the phages of lambda C that enters into them by the restriction endonuclease system, the endonuclease property of this restriction modification system. But in some of these cells, the phage gets successfully modified with the methylase activity of the same cell. So this restriction and modification, that is the endonuclease and methylase activities, they go side by side in a cell. So it is the property of the cell that it either will restrict the entry of the foreign DNA or will modify it so that it can replicate. By the same mechanism of modification, the host cell protects its own DNA from the activity of the endonuclease that is present within the cell, because bacterial cells don't have nuclear membrane and the DNA is naked. That means the restriction endonuclease has complete access to the DNA, the host DNA, but the host DNA has been methylated and modified and is protected by that manner by the restriction endonuclease that it harbors. Hamilton, Smith and Wilcox in the year 1970 discovered the first restriction endonuclease from Haemophilus influenzae strain RD. This enzyme is now known as Hindi 2. Shortly after that, many different restriction endonucleases were isolated and characterized, and we now know that there are thousands of different types of restriction endonucleases.